Welcome to the deep dive. Today we're taking a journey into, well, what might be the fastest, most complex, and most accurate assembly line out there. Bacterial genetics. We're decoding the ultimate biological code base, how bacteria copy, read, and execute their genetic instructions. We're drawing on some foundational sources in microbial biology for this.
That's right. Our mission today really is to walk through these mechanisms of inheritance and expression. We're looking at the core polymerization reactions that build DNA, RNA, and protein. It's the chemical heart of life.
It really is.
But to truly appreciate the system, you have to look at the scale. It's just mind-boggling.
And that scale is something scientists are actually starting to mimic now, isn't it? For millennia, DNA has been this perfect storage medium. It's stable, incredibly compact. It's like the original digital archive.
Exactly. And what's fascinating is that modern science is trying to catch up. They're experimenting with something called biostorage. They realize, the information in thousands of books could fit into a tiny, tiny fraction of a DNA strand.
Which they actually proved, right?
Yeah.
I remember reading about encoding a GIF, of all things.
Yeah, a little GIF of a horse and rider. They converted the binary code into A, G, C, T, synthesized that DNA, and then managed to store it stably inside E. coli.
Amazing.
It just shows life figured out the perfect hard drive billions of years ago. To really appreciate the genius of this code, we have to remember that for decades, scientists were actually pretty skeptical about DNA. It seemed, too simple.
Yeah, it's hard to believe now, but the argument was DNA only has 4 letters, 4 nucleotides. How could that possibly store all the complexity of life? Proteins, with their 20 different amino acids, seem like a much better bet.
Exactly. The first big hint that DNA was the key came from Fred Griffith's work in 1928. The famous transformation experiments with Streptococcus pneumonia.
Right, the smooth and rough strains in mice.
Precisely. He showed that if you heat-killed the virulent S strain, it couldn't cause disease. But if you mix that heat-killed S strain with live, non-virulent R strain bacteria, somehow the R strain became deadly. Something was transferred.
A transforming principle, he called it. But what was it?
That was the million-dollar question. Avery, McLeod, and McCarty tackled that in 1944. They took extracts from the S strain and systematically destroyed different components using enzymes, RNA, protein, or DNA.
And the key finding?
The key finding was that transformation, the ability to make those R cells virulent, was blocked only when they destroyed the DNA. Destroying RNA or protein had no effect. That was the first really strong piece of evidence pointing to DNA.
Okay, so strong evidence, but maybe not the final nail in the coffin.
Not quite definitive for everyone. That came later, in 1952, with the Hershey and Chase experiment. Classic stuff. They used a bacteriophage, a virus that infects bacteria.
And they used radioactive labels.
Right. Phosphorus-32, which labels DNA because DNA has phosphate groups, but proteins don't really. And sulfur-35, which labels protein because some amino acids have sulfur, but DNA doesn't.
Clever. So they let the virus infect the bacteria.
Then they used a blender, literally, like a kitchen blender, to shear off the virus particles stuck to the outside of the bacteria. Then they centrifuged it to separate the bacteria, the pellet, from the liquid, the supernatant, with the viral coats.
And the result?
Unmistakable. The sulfur-35, the protein label, stayed mostly in the supernatant, but the phosphorus-32, the DNA label, was found inside the bacterial pellet.
So the DNA went in. That was the genetic material.
Exactly. That really settled it. DNA carries the instructions.
Okay, let's unpack the structure of the code itself. DNA, everyone knows the double helix, right? 2 strands coiled up. What are the key rules governing this structure?
Well, the rules are all about elegance and polarity. First, the 2 strands are antiparallel. Think of them like 2 lanes of a highway running in opposite directions. One runs 5' to 3', the other runs 3' to 5'.
Got it. And they stick together.
How? Through complementary base pairing. Adenine A always pairs with thymine T, using 2 hydrogen bonds. Guanine G always pairs with cytosine C, using 3 hydrogen bonds. That GC bond is a bit stronger.
And this pairing holds the 2 strands together along the whole length.
Yes, and the backbone of each strand is made of deoxyribose sugar and phosphate groups linked by strong phosphodiester bonds. This whole structure creates those famous major and minor grooves, which are important for protein interactions, and it's incredibly stable.
So DNA is the stable master blueprint. What about RNA? It's related, but different.
Yeah, RNA is more like the working copy, or sometimes a functional tool itself. It uses a slightly different sugar, ribose, instead of deoxyribose, and instead of thymine T, it uses uracil U to pair with adenine A.
And it's usually single-stranded.
Often, yes. But the key thing about RNA is that even as a single strand, it can fold back on itself into really complex 3D shapes. Think of tRNA. Transfer RNA, its specific shape, is crucial for its job in translation.
Okay, back to DNA. You mentioned bacterial chromosomes are typically circular and closed. That sounds constrained.
It is. Imagine trying to store a really long coiled phone cord in a small box it naturally twists up on itself. That's supercoiling. Most bacterial DNA is negatively supercoiled.
Negative supercoiling. What does that mean?
It means the DNA is slightly underwound, sort of loosened up compared to its relaxed state. It sounds counterintuitive, but this actually makes it easier for the cell to separate the two strands when it needs to access the information for replication or transcription. It's a clever storage solution.
Right. So once we understand the blueprint structure, the next big question is, how does the cell copy this massive supercoiled document without making mistakes, especially at speed?
Exactly. DNA replication has to be incredibly fast. We're talking 750 to 1,000 base pairs per second in bacteria.
Wow.
And yet, it's astonishingly accurate. The error rate is something like 1 mistake in every billion or even 10 billion base pairs copied.
How is that even possible?
It uses what's called the semiconservative model. When the DNA duplicates, each new double helix consists of one of the original parental strands and one newly synthesized strand. So it keeps half the old, builds half the new.
And in bacteria, it all kicks off at one specific spot.
Usually, yes, a single origin of replication called auric. From there, 2 replication forks start moving out in opposite directions, going around the circular chromosome. If you could see it, it would look kind of like the Greek letter theta as it progresses.
Okay, so 2 forks moving super fast. What's actually doing the work at these forks?
It's a whole team of proteins, a complex machine called the replisome. First, you need something to unwind the double helix, that's helicase, often called DNAB in E. coli. It uses ATP energy to pry the strands apart.
And once they're apart, they don't just snap back together.
Nope, because single-stranded binding proteins, or SSBs, immediately coat the separated strands to keep them from re-annealing.
Okay, but wait. If you're unwinding a closed circle that fast, wouldn't the DNA ahead of the fork get incredibly tangled and wound up like twisting a rope?
Absolutely. You get massive torsional stress and positive supercoiling building up ahead of the fork. That's where topoisomerases come in. In bacteria, a key one is DNA gyrase.
What does it do?
It's like a molecular stress reliever. It makes temporary cuts in the DNA backbone, allows the strands to rotate around each other to release the tension, and then seals the cuts back up. It prevents the whole process from seizing up. Essential.
Right, so helix open, tension managed. Now, who actually builds the new DNA strand?
That's the job of the main enzyme, DNA polymerase III holoenzyme. It reads the template strand and adds the correct complementary nucleotides to the new strand, synthesizing it in the 5-3-1 direction.
But I remember something about polymerase needing a starting point. You can't just begin from scratch.
Correct. DNA polymerases need a free 3' hydroxyl group, dialo-H, to add onto. They can only extend an existing chain. So another enzyme called primase, which is actually a type of RNA polymerase, lays down a short RNA primer, maybe 10 nucleotides long. That provides the necessary starting point for DNA pol 3.
Ah, okay. But DNA pol 3 only works in one direction, 5-3 pick, and the 2 template strands are antiparallel. How does that work at the fork?
That creates a bit of a puzzle, and the cell solves it elegantly, if a bit complicatedly. This leads to the famous leading and lagging strands.
Explain those.
Okay, so one template strand, the one running 3 to 5 foot towards the fork, allows the new strand to be synthesized continuously, also moving towards the fork. That's the leading strand. Smooth sailing. Needs just one primer to get started.
Makes sense.
But the other template strand is running 5 to 3 foot towards the fork. Since DNA pol 3 can only synthesize 5 to 3 foot, it has to build the new strand away from the fork's movement.
So it has to keep starting over.
Exactly. It synthesizes a short piece, then the fork moves further, primase lays down another primer, and pol the third makes another short piece. These short, discontinuous fragments are called Okazaki fragments. They're about 1,000 to 3,000 nucleotides long in bacteria. This is the lagging strand.
So you end up with a new strand made of lots of little DNA pieces mixed with RNA primers. That needs cleaning up.
It does. Now another player steps in. DNA polymerase is first. It has a special ability of 5 to 3 foot exonuclease activity that lets it chew away the RNA primers. As it removes the RNA, it simultaneously fills the gap with DNA nucleotides.
Almost there. But there must be tiny gaps left between the fragments where polymerase finished.
Precisely. Little nicks in the sugar phosphate backbone. The final step is DNA ligase, which seals these nicks, creating the last phosphodiester bond and making the lagging strand whole and continuous. It uses energy, often from NAD+ in bacteria.
Okay, that explains the mechanics. But what about that incredible accuracy? 1 in 10 billion errors.
That's largely due to the proofreading capability built right into DNA polymerase III. It has a subunit, the epsilon subunit, that acts like a backspitz key. If the polymerase accidentally adds the wrong nucleotide, this 3 to 5' exonuclease activity immediately senses the mismatch, pauses, removes the wrong base, and lets the polymerase try again. It catches most errors right on the spot.
Amazing. So replication proceeds around the circle.
Yeah.
How does it stop?
For most circular chromosomes, like E. coli's, there are specific termination sites called TER sites on the opposite side of the chromosome from ORIC. A protein called TUS binds to these sites and physically blocks the helicase, halting the replication forks when they meet there.
And what about bacteria with linear chromosomes, like Lyme disease bacteria? They must have an issue at the ends, like our chromosomes do.
They do face an end replication problem, yeah. Bacteria like Borrelia burgdorferi have evolved really interesting solutions. They often have covalently closed hairpin ends on their linear DNA, and they use specialized enzymes like telomere resolvase REST to resolve and separate the duplicated chromosomes without losing genetic information from the tips. Quite different from the circular model.
Okay, so we've copied the DNA blueprint. Now, how does the cell actually read the instructions encoded within it? This brings us to genes and transcription, right?
Exactly. A gene basically is a stretch of DNA that codes for a functional product. That product is often a protein, but it can also be functional RNA molecules like transfer RNA, tRNA, or ribosomal RNA.
And getting from the DNA gene to that product involves making an RNA copy first.
Yes. That process is transcription, synthesizing an RNA molecule using a DNA template.
How are bacterial genes typically organized? I've heard they're very efficient.
They are incredibly efficient. Often, genes with related functions say all the enzymes needed for a specific metabolic pathway are clustered together on the chromosome and controlled by a single switch. This cluster is called an operon.
And the whole cluster gets transcribed together.
That's the beauty of it. One promoter region controls the transcription of the entire operon, resulting in a single long messenger RNA, mRNA molecule that contains the coding sequences for multiple proteins. This is called a polycistronic mRNA.
That saves a lot of regulatory overhead.
Absolutely. Then, during translation, ribosomes can initiate synthesis at multiple points along that single mRNA to make all the different proteins needed for that pathway simultaneously.
Okay, let's look at a typical protein coding gene within an operon, or even one that stands alone. What are the essential parts?
You always start with the promoter. This is the DNA sequence upstream of the actual coding part, and it's where the transcription machinery binds. It's not transcribed itself. Key parts of the promoter in bacteria include consensus sequences around the -35 and -10 positions relative to the start of transcription. The -10 region is often called the Pribnow box.
So the promoter is the start here signal. What comes next?
After the promoter, there's usually a leader sequence. This part is transcribed into RNA but doesn't get translated into protein. Crucially, in bacteria, the leader contains the Shine-Dalgarno sequence.
Shine-Dalgarno. That's important for translation later, right?
Critically important. We'll come back to that. After the leader comes the actual coding region. This starts with a specific start codon, usually AUG, and ends with one of 3 stop codons, UAA, UAG, or UGA. This is the part that dictates the amino acid sequence of the protein.
And after the stop codon?
There's usually a trailer sequence, again transcribed but not translated. And finally, you have the terminator sequence, which signals the transcription machinery to stop and release the newly made RNA molecule.
Okay, so who's the main player doing the transcribing?
That's RNA polymerase. In bacteria, the core enzyme has several subunits, 2 alpha, beta, beta primors, and omega. This core enzyme is responsible for actually synthesizing the RNA chain.
But you mentioned the promoter is the binding site. How does the core enzyme know which promoter to bind to? It can't just randomly start anywhere.
It doesn't. It needs help. That's where the sigma factor comes in. Sigma is a separate protein, a transcription factor that binds to the core enzyme. This combination forms the holoenzyme.
So holoenzyme equals core plus sigma.
Right, and it's the sigma factor's job to recognize those specific NENIC35 and NENIC10 sequences in the promoter. So the holoenzyme specifically targets promoters, while the core enzyme alone can't initiate transcription properly.
And bacteria have different sigma factors.
Yes, quite a few. The main housekeeping one is usually sigma 70, which recognizes most standard promoters. But there are alternative sigma factors that recognize different promoter sequences, allowing the cell to turn on specific sets of genes in response to different conditions, like heat shock or nutrient starvation. It's a major way they regulate gene expression globally.
Okay, so walk us through the transcription cycle itself. Initiation, elongation, termination.
Sure. Initiation starts when the RNA polymerase Hall enzyme binds to the promoter, forming a closed complex. Then, the polymerase unwinds the DNA locally, usually around the -10 region, which is AT-rich, easier to melt, forming an open complex. Now it can start synthesizing the RNA chain using one DNA strand as a template. After about 12 or so nucleotides are joined, the sigma factor usually dissociates. Its job is done.
So the core enzyme carries on alone now.
Yes, that's elongation. The core RNA polymerase moves along the DNA template, unwinding it ahead and rewinding it behind, synthesizing the RNA molecule complementary to the template strand, remembering U pairs with A. And just like in replication, topoisomerases are working nearby to relieve any supercoiling stress caused by the unwinding.
And finally, it has to stop. Termination. You mentioned terminator sequences. How do they work?
There are two main mechanisms in bacteria. The most common is factor-independent termination, sometimes called intrinsic termination. The DNA sequence in the terminator region contains an inverted repeat, followed by a string of adenines.
Inverted repeat.
Meaning a sequence followed shortly by its reverse complement. When this gets transcribed into RNA, the inverted repeat allows the RNA to fold back on itself, forming a stable hairpin loop structure, like a stem with a loop at the end. This hairpin structure forms right behind the polymerase. Right after the hairpin, the RNA is paired with the DNA template in that run of A's, meaning the RNA has a run of U's, UA pairs. UA pairs are the weakest RNA-DNA interaction. The formation of the hairpin seems to destabilize this weak pairing, essentially causing the RNA polymerase to pause and then just fall off the DNA template, releasing the RNA transcript.
Clever. And the other mechanism?
The other one is rho-dependent termination. This requires a protein factor called rho. Rho recognizes and binds to a specific sequence on the growing mRNA transcript called the RUT site, Rho utilization site.
So it binds the RNA, not the DNA.
Correct. Rho then uses ATP energy to move along the mRNA, chasing after the RNA polymerase. If the polymerase pauses at a specific rho-dependent termination site, often a sequence that causes pausing, Rho catches up. It has helicase activity, meaning it can unwind nucleic acid hybrids. So when Rho reaches the paused polymerase, it actively separates the RNA transcript from the DNA template, causing termination.
Two different ways to hit the brakes. Seems efficient.
Right. So now we have our messenger RNA transcript. The next and final major step is translation. Decoding that mRNA sequence into a specific sequence of amino acids, building a protein. This is where the genetic code truly gets read.
And the code is read in triplets, right? Codon?
Exactly. 3 consecutive nucleotides on the mRNA form a codon, and each codon specifies either a particular amino acid or a signal to start or stop translation.
So many codons are there?
Well, with 4 bases, A, U, G, C, read 3 at a time, there are 4 by 4 by 4 equals 64 possible codons.
And there are only 20 common amino acids.
Right. So most amino acids are specified by more than one codon. This is known as the redundancy or degeneracy of the genetic code. For example, leucine is specified by 6 different codons. Only methionine and tryptophan have just 1 codon each.
Does this redundancy matter?
It does. It provides some buffer against mutations. A change in the DNA might change the codon, but still specify the same amino acid. Also, it relates to something called the wobble hypothesis.
Okay, what's the wobble hypothesis? Sounds imprecise.
It's actually a very precise kind of imprecision. It saves the cell resources. See, the codons on the mRNA are recognized by transfer RNAs, tRNAs, which carry the corresponding amino acid. Each tRNA has an anticodon loop that base pairs with the mRNA codon.
Okay.
The wobble hypothesis, proposed by Francis Crick, states that the base pairing between the third position of the mRNA codon, the 3' base, and the first position of the tRNA anticodon, the 5' base, doesn't have to be as strict as the standard AU and GC rules. There's some wobble allowed.
For example?
For example, a U in the wobble position, the tRNA anticodon, can pair with either A or G in the third position of the mRNA codon, or a G can pair with U or C. This means that a single tRNA species can often recognize multiple codons that code for the same amino acid.
Ah, so the cell doesn't need 61 different tRNAs for the 61 sense codons. It can get by with fewer.
Exactly. It minimizes the number of tRNA genes the cell needs to have and maintain. Very efficient.
So these tRNAs are the crucial adapters, linking the nucleic acid code to the amino acid sequence. What do they look like?
In 2D, they're often drawn as a cloverleaf structure, with several loops and stems formed by base pairing within the single RNA strand. In 3D, they fold into a compact L shape. Two key parts are the anticodon loop at one end, which reads the mRNA codon, and the acceptor stem at the other end, specifically the 3' CCA sequence, where the correct amino acid gets attached.
And how does the correct amino acid get attached? That seems critical for accuracy.
Absolutely critical. This is done by a set of enzymes called aminoacyl-tRNA synthetases. There's generally one specific synthetase for each amino acid. These enzymes perform a two-step reaction using ATP energy to charge the tRNA, meaning covalently link the correct amino acid to its corresponding tRNA molecule. They are incredibly specific. They have proofreading mechanisms, too, to ensure the right amino acid is attached to the right tRNA. Mistakes here would be disastrous.
Okay, so we had the mRNA message and the charged tRNAs ready to bring the amino acids. Where does the actual protein synthesis happen?
On the ribosome. This is the massive molecular machine, the workbench for protein synthesis. In bacteria, it's the 70S ribosome, composed of two subunits, the small 30S subunit and the large 50S subunit. Both subunits are made of ribosomal RNA, rRNA, and ribosomal proteins.
And the rRNA is just structural.
Not at all. The RNA is actually the functional core. Particularly, the 23S RNA found in the large 50S subunit acts as the enzyme that catalyzes the formation of peptide bonds between amino acids. It's a ribozyme, an RNA molecule with catalytic activity. The proteins are important for structure and stability, but the RNA does the heavy lifting of catalysis.
Wow, and the small subunit.
The 16S RNA in the small 30S subunit plays a crucial role in initiation. Remember that Shine-Dalgarno sequence on the mRNA leader?
Yeah.
The 16S RNA has a sequence that is complementary to the Shine-Dalgarno sequence. This base pairing interaction correctly positions the mRNA on the small subunit so that the start codon, usually AUG, is placed precisely in the right spot for translation to begin. This ensures the ribosome starts reading in the correct frame.
Okay, so let's walk through the process. Initiation first.
Initiation involves assembling the whole complex. The 30S subunit binds to the mRNA via Shine-Dalgarno 16S rRNA. Then, a special initiator, tRNA carrying N-formylmethionine, fMet in bacteria, binds to the start codon, AUG, which has been positioned on what's called the P site, peptidyl site, of the ribosome. Several initiation factor proteins help this process. Finally, the large 50S subunit joins the complex, completing the 70S initiation ribosome, ready for elongation.
So the first tRNA starts in the P site. What about the other sites? I think there are A and E sites too.
Right. The ribosome has three main sites for tRNAs. The A site, aminoacyl site, the P site, peptidyl site, and the E site, exit site.
So the initiator tRNA is in P. What happens next in elongation?
Elongation is a cycle. Step 1, aminoacyl-tRNA binding. The next charged tRNA carrying the amino acid specified by the second codon on the mRNA enters the A site. This requires help from elongation factors like EF2 and energy from GTP hydrolysis.
So now you have tRNA in P and tRNA in A.
Step 2, transpeptidation. This is the core reaction. The 23S rRNA, the peptidyl transferase center, catalyzes the formation of a peptide bond. It transfers the amino acid, or the growing polypeptide chain, from the tRNA in the P site onto the amino acid attached to the tRNA in the A site. The energy for this bond comes from the high-energy bond linking the amino acid to the tRNA in the P site.
So the polypeptide chain moves to the tRNA in the A site.
Exactly. Now the tRNA in the P site is empty, and the tRNA in the A site carries the growing polypeptide chain. Step 3, translocation. The entire ribosome moves exactly one codon down the mRNA towards the 3' end. This shift requires another elongation factor, EFG, and more GTP hydrolysis.
What does that shift do to the tRNAs?
The tRNA carrying the polypeptide chain, which was in the A site, moves into the P site. The now empty tRNA, which was in the P site, moves into the E site. And the A site is now empty again, positioned over the next codon, ready to accept the next incoming charged tRNA.
And the empty tRNA in the E site just leaves.
Yep, it exits the ribosome, and the cycle repeats. tRNA binding to A, peptide bond formation, translocation. Over and over, adding one amino acid at a time, reading the mRNA 5' to 3 minute, and building the protein from its N-terminus to its C-terminus.
That sounds energy-intensive. GTP at multiple steps.
Very. Protein synthesis is one of the most energy-consuming processes in the cell, but bacteria have ways to make it super efficient. For one, a single mRNA molecule is usually translated by multiple ribosomes simultaneously. As soon as one ribosome clears the start codon, another one can hop on. This structure, an mRNA with multiple ribosomes attached, is called a polyribosome or polysome.
So you get many protein copies from one mRNA very quickly.
Exactly. And even more remarkably, in bacteria, because there's no nucleus separating transcription and translation, these processes are often coupled, meaning ribosomes can actually bind to the 5' end of the mRNA and start translating it while the RNA polymerase is still transcribing the rest of the gene further downstream.
Wow, that's incredibly efficient. No waiting around.
None at all. It maximizes the speed of gene expression.
Okay, so elongation continues
Until the ribosome encounters one of the 3 stop codons, UAA, UAG, or UGA, in the A site. There are no tRNAs that recognize these codons. Instead, proteins called release factors, RFs, recognize the stop codons.
And what do?
Binding of a release factor, like RF1 or RF2, depending on the codon, plus RF3, triggers the hydrolysis of the bond linking the completed polypeptide chain to the tRNA in the P site. The polypeptide is released, and then other factors help the ribosomal subunits, the mRNA, and the empty tRNA to dissociate, ready to start the process again. That's termination.
So the polypeptide chain is released, but it's just a linear string of amino acids at this point, right? It's not a functional protein yet?
Correct. It needs to fold into its precise, unique, three-dimensional shape to become functional. And sometimes, especially in bacteria, the N-terminal methionine, or fMet, needs to be processed, like removing the formal group or even the entire methionine.
Folding seems tricky. How does it find the right shape out of all the possibilities and avoid clumping up?
It is tricky, especially in the crowded environment of the cytoplasm. Misfolded proteins can aggregate and become toxic. That's why cells have molecular chaperones. These are proteins that help other proteins fold correctly or prevent them from folding incorrectly or aggregating.
What are some examples in bacteria?
Well, one important one acts very early, trigger factor, TF. It associates directly with the ribosome near the exit tunnel where the polypeptide emerges. It can bind to hydrophobic patches on the nascent chain, preventing them from misfolding or sticking together while the protein is still being synthesized.
So it protects it right from the start. What about after it's released?
Then other cytoplasmic chaperone systems can take over if needed. A major one involves DNAC, related to HSP70 in eukaryotes, DNAJ, HSP40, and GRPE. This system uses ATP energy to bind to unfolded or misfolded proteins and help them reach their proper conformation.
And I've heard of another one, like a protein folding cage.
Ah, you mean the GRO-L-GRO system. This is really remarkable. GRO-EL forms a large barrel-shaped complex with a central cavity. Misfolded proteins can enter this cavity. Then a lid complex, GRO-ES, caps the barrel. Inside this protected chamber, using ATP hydrolysis, the chaperone helps the protein unfold and then refold correctly, shielded from the crowded cytoplasm. It's essential for folding some complex or aggregation-prone proteins.
Okay, so proteins are folded, but many bacterial proteins need to end up outside the cytoplasm, right? In the membrane or the periplasm, or even completely outside the cell.
Absolutely. We need to distinguish between translocation, moving a protein across or into the plasma membrane, and secretion, which usually implies moving a protein completely out of the cell. In gram-negative bacteria, secretion means crossing both the inner and outer membranes.
Are there common systems for just getting across that first barrier, the plasma membrane?
Yes, there are 2 major pathways found in nearly all bacteria for getting proteins across the inner membrane. The main workhorse is the CO system, or the general secretion pathway. It primarily translocates proteins in an unfolded state.
Unfolded. How does it work?
Proteins destined for sec transport have a special N-terminal signal peptide. This signal targets the protein either post-translationally via the secamotor protein, which uses ATP, or co-translationally via the signal recognition particle, SRP pathway, to a channel in the membrane formed by sexy, sexy-E, and sexy proteins, the sexy translocon. The unfolded protein is threaded through this channel using energy from both ATP hydrolysis, by secas, and the proton motive force, PMF, across the membrane. The signal peptide is usually cleaved off after translocation.
Okay, sexansyl unfolded proteins. What if a protein needs to fold in the cytoplasm first? Maybe because it incorporates a cofactor before it gets transported.
That's where the second major pathway comes in, the TAT system, which stands for twin arginine translocase. This system is specifically designed to transport fully folded proteins across the plasma membrane.
Folded proteins, how does it get something that bulky across?
It's pretty amazing. TAT substrates have a special signal peptide containing a pair of arginine residues, hence the name twin arginine. The exact mechanism is still being studied, but it involves assembling a large translocon complex in the membrane. Remarkably, it seems to transport these folded proteins using only the energy stored in the proton motive force, PMF. No ATP required directly.
So sex or unfolded, TAT folded across the inner membrane. What about getting things all the way out, especially in gram-negatives with that outer membrane?
Gram-negative bacteria have evolved a whole arsenal of specialized secretion systems, often called type 1, type 2, type 3, and so on, up to type 7 and beyond in some cases. Many of these are one-step systems, meaning they form a continuous channel spanning the entire cell envelope, inner membrane, periplasm, and outer membrane.
Give us a couple examples. What are type I and type 3 like?
Type I secretion systems, T1SS, are relatively simple. They consist of three main components: an ABC transporter in the inner membrane that provides energy, a membrane fusion protein that bridges the periplasm, and a channel-forming protein in the outer membrane. They often transport toxins or enzymes directly from the cytoplasm to the outside in one go.
Okay. And type 3? I've heard that one called an injectisome.
That's right. Type 3 secretion systems, T3SS, are famous, particularly in pathogens. They form a structure that looks remarkably like a molecular syringe or needle, evolutionarily related to the base of the bacterial flagellum.
The syringe, what does it inject?
Pathogens use T3SS to inject bacterial effector proteins directly from their cytoplasm into the cytoplasm of a host eukaryotic cell. These effectors then manipulate the host cell's functions to the bacterium's advantage, maybe suppressing the immune response or forcing the host cell to take up the bacterium. It's a key virulence mechanism.
Wow. Direct injection. Are there others like that?
Well, there's also the type beta-6 secretion system, T6SS. This one is also like a weapon, but it's more like a contractile dart gun. It's structurally homologous to parts of bacteriophage T4, specifically the tail sheath and tube.
A contractile dart gun. What's it firing?
It fires effector proteins, often toxins, into adjacent cells. This can be used for bacterial warfare, killing competing bacteria, or sometimes for interacting with host cells. The T6SS literally contracts its outer sheath to propel an inner tube tipped with effector proteins across the membranes and into the target cell. It's incredibly dynamic.
These secretion systems sound like sophisticated nanomachines. Is that the only way bacteria export things?
Not quite. There's another fascinating mechanism: membrane vesicles. Bacteria can actually bud off small spherical vesicles from their membranes.
Like little packages.
Exactly. Gram-negative bacteria shed outer membrane vesicles, OMVs, which pinch off from the outer membrane. Gram-positive bacteria lacking an outer membrane release extracellular vesicles, EVs, that bud from their plasma membrane and somehow make it through the thick peptidog-lichen wall.
What's inside these vesicles?
All sorts of things. They can contain outer membrane proteins, periplasmic contents, toxins, enzymes, signaling molecules, even DNA or RNA. It's basically a way to deliver a concentrated package of molecules protected by a lipid bilayer to other cells or the environment. They're involved in communication, nutrient acquisition, stress response, and pathogenesis. A whole other layer of interaction. So if we just take a step back and look at the whole picture, from copying the DNA with incredible fidelity to transcribing the messages to translating them into proteins on the ribosome and then folding and delivering those proteins, the energetic cost is just immense.
Yeah, all that ATP and GGP used at almost every step.
Exactly, especially translation charges in the tRNAs, initiation, elongation factor activity. It consumes multiple high-energy phosphate bonds for every single amino acid added to the chain. This huge investment really underscores why accuracy and efficiency are paramount. The cell cannot afford to waste energy on sloppy synthesis or making useless proteins.
It really is an engineered marvel when you think about it that way. The stable DNA blueprint gets copied nearly perfectly, the transient RNA messages are made, and read using clever tricks like operons and the wobble hypothesis.
And then these complex protein machines are built, meticulously folded with the help of chaperones, and delivered precisely where they need to go, using systems ranging from simple sec channels to these incredibly complex, weaponized secretion systems like T3SS and T6SS.
Yeah, that complexity is just staggering, especially when you remember what you said earlier, how scientists initially thought DNA was too simple. They completely missed the layers upon layers of intricate machinery needed to actually use that code.
Absolutely. The coordination required for all of this to happen simultaneously, correctly, and rapidly within a single tiny bacterial cell is just mind-blowing.
So maybe a final thought for our listeners. When you think about bacteria, maybe especially pathogens, consider those injection systems, the T3SS injectisome, the T6SS dart gun. This genetic code we've been discussing, it's not just the blueprint for building the cell itself, it's also the blueprint for building the weapons and tools these microbes use to compete, to survive, and to interact with their environment, including us. How do these nanomachines, encoded by just a handful of genes, fundamentally shape microbial communities, drive virulence, and ultimately impact our own health. That microscopic arms race, driven by this code, is definitely worth a deep dive.